Hello everyone. Today we'll learn about virulence and efficacy of pesticides. Virulence refers to the nature and extent of a pesticide's lethal effects on control targets, that is, the degree of harm it can cause to harmful organisms. The virulence of various pesticides is evaluated under strictly controlled conditions, using precise testing methods with standardized test insects, pathogens, or weeds as benchmarks. Efficacy is built on virulence. Only when the virulence is proven high in lab tests can it be applied in the field. Here are the basic conditions for testing pesticide virulence. 1. Standard test pesticides. 2. Standard test organisms. 3. Standard test conditions. 4. Standard test methods. Median lethal dose, LD50, or median lethal concentration, LC50, is the dose, LD50, or concentration, LC50, of pesticide required to kill 50% of a specific biological population. The smaller the value, the more toxic the pesticide is to the insects. Median effective dose, ED50, or median effective concentration, EC50, is the dose or concentration required to inhibit the germination of 50% of fungal spores. Efficacy refers to a pesticide's effect on harmful organisms. It is often tested under natural outdoor conditions. Efficacy and virulence are usually closely related. The higher the virulence, the greater the efficacy. However, the pesticide's quality, environmental conditions during testing, plant growth status, and the application methods are also critically important and should be considered when testing. Here are the basic conditions for testing pesticide efficacy. 1. The test materials and soil fertility are roughly the same. 2. The field is heavily infested with pests, diseases, and weeds. 3. Proper field management systems and suitable climatic conditions. 4. Correct experimental design. Methods for expressing pesticide efficacy can vary with the pesticide's type and its control targets, but the underlying principle is similar. Insecticide efficacy is often shown as a mortality rate. Mortality rate is a fundamental measure of the efficacy of an insecticide. It's the percentage of the insects killed after treatment relative to the total number of test insects. However, natural deaths often occur in the control groups treated with different pesticides, so correction is needed in field control trials. Pest density or damage levels are typically measured after treatment, using indicators like the number of surviving insects or the percentage or quantity of population increase or decrease. Insect population density and insect population reduction rate are the most commonly used indicators. Biocide efficacy is typically measured based on the type of disease and its severity, such as incidence rate, disease index, and efficacy, with efficacy expressed as the changes in dry or fresh weight of weeds. The efficacy of chemical pesticides in the field depends on the interaction between the pesticide, the organism, and the environment. Pesticide-related factors. Pesticides with different active ingredients vary in their efficacy. Their physical and chemical properties, such as solubility, wetting ability, spreadability, dispersibility, and stability, all affect their efficacy. Different mechanisms and modes of action lead to different efficacy. Stomach insecticides are effective against chewing pests, but not so against piercing, sucking ones. Different concentrations or dosages also result in different efficacy. Typically, higher concentrations lead to higher efficacy, but after a certain point, higher concentrations may not lead to higher efficacy. If too much a pesticide is applied, it can kill the conducting tissues, preventing the pesticide from reaching the roots or growth points, resulting in poor efficacy. Applying smaller doses multiple times works better. Excessive pesticide use can also lead to waste without boosting its efficacy. It can easily cause phytotoxicity to plants, promote rapid resistance development in pests, kill natural predators, and pollute the environment. Every living organism has its weak spots, making it vulnerable to external factors such as pesticide attacks or interference, which can threaten its continued existence. Knowing these weak points is crucial for the proper and scientific use of pesticides. 1. Pesticide efficacy varies among insects with different feeding mechanisms. Stomach insecticides sprayed on crops, leaves, stems, and fruits, or scattered as toxic baits across the field, are highly effective against chewing pests, but not against piercing, sucking ones. 2. Pesticide efficacy varies at different stages of the pest's life cycle, with control most effective before the third instar. In production, the best time for control is 1 to 5 days following the peak of egg hatching. In disease control, consider the pathogen's physiological state. Early treatment is most effective for plant diseases. When it comes to annual weed removal, herbicides should be applied during germination, or no later than when they have 3 to 5 leaves. For perennial weed removal, herbicides should be applied before flowering. 3. Pesticide efficacy varies with the pest's physiological state. Female pests typically exhibit greater resistance than males. In addition, environmental conditions affect not only the volatility, degradation, and other physicochemical properties of a pesticide, but also organisms' absorption, detoxification, and poisoning processes. Similarly, temperature, humidity, and other conditions can also influence the pesticide's efficacy. There are some measures for improving pesticide efficacy. 1. Carefully consider the control targets, crop species, and their growth stages to choose the right pesticide. 2. Consider the growth stages of diseases, pests, and weeds, the growth status of crops, and the quality of the pesticide to ensure timely and effective treatment. 3. Apply a pesticide on sunny mornings or evenings. Avoid application during rainy weather, strong winds, or high temperatures. Calculate the application amount for the field based on the recommendations without altering it arbitrarily. 4. Ensure proper mixing of pesticides to enhance control efficacy, extend the coverage, reduce costs, and boost safety for humans and animals. 5. Choose proper application methods and machinery to ensure the pesticide is evenly applied to the crops or pests. 6. Strengthen management with a focus on prevention. alternate or mix different pesticides, or apply them intermittently.